Hi, good afternoon, everyone. My name is Jenny Wang. Today, the topic I'm going to talk about is approaches to analyzing comparative use human factor studies. So this is a disclaimer. So the learning objectives for my presentation today, the first one is to provide the overview of the current use of comparative use human factor studies to support other design differences. And the second objective is to discuss the analysis approach for the non-inferiority test in comparative use human factor studies. First, I will talk about the current use of human factor studies to support other design differences. So in the FDA's draft guidance, the comparative analysis and the related comparative use human factor studies for a drug device combination product submitted in an ANDA, we can see like in this procedure at the very beginning, during the early stage of the development, applicants are encouraged to minimize the differences from the user interface for the reference for the RLD product. And then applicants can conduct the threshold analysis, including labeling comparison, comparative task analysis, and the physical comparison of the delivery device constituent part. So the result from the threshold analysis may be no design differences or differences in design. For the differences in design, if that falls into the categories that's other design differences, applicants can either modify the user interface to minimize the differences or to provide data to support the differences, just as my colleague just presented. Applicants can provide, can conduct a comparative use human factor studies to support the differences. So for the human factor studies, it's designed to confirm that the use error rate for the critical task for the proposed generic combination product is not worse than the corresponding use error rate for the RLD. So here is a procedure of comparing the error rate of test product and the error rate of the RLD product through the non-inferiority test in human factor studies. There are mainly like 4 steps. The first step is to determine the allowable margin D by which the error rate of the test product could exceed the error rate of RLD product. The second step is to estimate the study sample size, considering assumed error rates and the margin D. The third step is to observe error rate for the critical task during the human factor experiments. And the fourth step is to perform the NI hypothesis test. So I will introduce the details of each step in the following slides. So for the first step is to determine the allowable margin D by which the error rate of the test product could exceed the error rate of RLD product. So the value of this margin D will differ between products. It's depending on the indications and the clinical consequences associated with failing to perform the critical tasks appropriately. And the acceptable margin D should be decided in consultation with FDA before the study is conducted. The second step is to calculate the study sample size, considering assumed error rates and the margin D. So in the FDA draft guidance, it provides an example using the Tango method to calculate some power simulations given selected sample size and the alpha level equals to 0.05 and the allowable margin D equals to 12.1, just as this table shows here on the right side. So the Tango method used in this example is the widely used method to calculate confidence intervals for the differences of two proportions in a paired design of clinical trials. To utilize the Tango method to calculate the confidence intervals, usually there are several parts of information required, like the number of subjects who completed the R tasks successfully but had errors in the T tasks, and the number of subjects who completed T tasks successfully but had errors in R tasks. Also, you will need to have the total number of subjects and the confidence level. We want to note here is that the Tango method is just one of the options for the analysis of the human factor studies. So the third step in this procedure is to observe error rates for the critical tasks during the experiment. To conduct the experiment, first of all, we will need to define the critical tasks. And then we will need to observe the error or success results of subjects for each critical task. Like on the right side is a table here. This is an example data set for a given critical task. We can see like each row can represent the result for each subject. So the first column represents the results for the RLD product, and the second column represents the results for the test product. We may use like this kind of binary results, like with the 1 means like subjects complete this task successfully, and the 0 means the subject may have encountered some errors or failed this task. So after the experiment, the fourth step is to perform the non-inferiority test. To conduct this non-inferiority test, we will need to, based on the experimental data, we will need to calculate the confidence intervals for the difference of error rates between the test product and the RLD product. Then we need to compare the upper bound of this confidence interval to the allowable margin D. So if given the alpha level equals to 0.05 and the upper bound of the 95% confidence interval is less than the margin D, then the null hypothesis is rejected and the non-inferiority is demonstrated. So for the data analysis part for the comparative use human factor studies, in addition to these current recommendations in the draft guidance, FDA continues to conduct internal research to facilitate drug development and mitigate regulatory burdens for the human factor studies. For example, like our group has been working on examining like the potential alternative data analysis methods and approaches for the human factor studies. We are working on evaluating the performance of different alternative methods. So the applicants are also encouraged to propose alternative data analysis methods and the study designs for the human factor studies. So here, there are just like some examples of the alternative data analysis methods to consider. For example, non-parametric methods may be considered, like the bootstrap-based methods, or Bayesian methods can also be considered to estimate the distribution of the target population's performance with the test and the RLD products. Applicants are also advised to discuss proposed alternative data analysis methods and study designs with FDA before initiating the comparative human factor studies. There are programs available for the discussions, like the Model Integrated Evidence Industry Meeting Pilot. So this is a new pilot program to offer meeting opportunities to applicants who intend to use model integrated evidence or novel data analytic approaches for BE establishment in their ANDAs. Also, the pre-ANDA program for complex generic products also appropriate program for the discussions. The ANDA applicants for complex generic drug products can request product development pre-ANDA meetings to help clarify regulatory expectations early in their product development stage. Here, I would like to acknowledge the help and the support from my colleagues and the leadership from the Office of Research and Standards of OGD. Thank you. So here is our first challenge question. So in the FDA draft guidance, comparative analysis and related comparative use human factor studies for drug-device combination products submitted in ANDA, the comparative use human factor study is recommended for every application of drug-device combination products. So this statement is true or false? Yeah, it's false. That's good news. So the second challenge question is, in the FDA draft guidance, so which of the following analysis methods is recommended for the non-inferiority test in comparative use of human factor studies? We have some options. First is Tango method. Second is bootstrap method. Third is Bayesian method. And the option D is the guidance doesn't recommend the analysis method for an eye test. Yeah, the correct answer is D. So Tango method was just used like in an example in the guidance. It's only one of the options for the analysis. Applicants encouraged to propose like novel analysis methods for human factor studies. And the FDA are open for discussions. Yeah, now we will go to the Q&A session. I would like to welcome Christina and our other panelists to join us on the stage. Thank you.